Each of the sausages composed of those protein molecules of lamins. So each of them identical to each other. And there are a certain type of mutation. Most of this mutation are the point mutation. A single amino acid substitution, sometimes frame shift mutation, several amino acid substitution, which cause the development of any of these two group of disorders. Would it be muscular dystrophy or premature aging? Specific kind of amino acid substitution would lead to either of these two. And so, therefore, some mutation in the lamin will cause the muscular dystrophy. The other mutation in a different position would cause the premature aging. Now, the second gene and the protein mutated in people suffering from these two disorders is a protein which is called amarin. Right? So when we talk about amarin, it's very important for you guys to remember how the sausages, these lamin filaments, how they actually attach to the inner membrane of the nucleus, right? So first of all, they are not attached physically by themselves. There is no direct contact between the inner membrane of the nucleus and any of the sausages, any of these nuclear filaments, right? They are attached with the help of a certain type of anchor. And these anchors are shown here, one anchor and another anchor. Each of them is also a protein molecule. And the name of these protein molecules are amarin and LBR. They have nothing to do with Roosevelt. It's a lamin binding receptor, right? A protein which LBR as well as amarin, they by themselves insert their bodies, their polypeptide chain into the inner membrane of the nucleus. All right. The first part is accomplished. And then they use also a certain portion of their polypeptide chain in order to bind these different sausages, these nuclear lamin filaments, right? And because these lamin filaments are attached to this LBR, lamin binding proteins, and amarin protein, these nuclear lamins are always located very close to the inner membrane, anchored, right? Now, then these nuclear lamina are also attached to the DNA molecules, different portions of different DNA molecules, and those portions of the DNA, so that would be a DNA, right? You see, some of the portions, some of the genes located in this nuclear DNA are attached or form a certain type of binding to these sausages, to this lamin filaments. And those regions are inactive. Their transcription is inactivated. They are silent, right? And then there are also some portions of the DNA, these white portions of the DNA, which are not attached to, which are not bound to the nuclear lamin. And they are transcribed. They are actively transcribed. So in other words, what kind of gene, a particular gene, is attached to this nuclear lamina would define whether or not this gene is silent, if it's attached and it's silent, or if a certain gene is not attached to the nuclear lamina, it will be transcribed actively, right? So in other words, the attachment to the nuclear lamina of DNA is used in order to shut down the transcription of a certain portion of the genes. And because, as I say, for instance, when you consider epithelial cells, then in epithelial cells, which look very specifically, you would recognize them immediately in a microscope and distinguish them, especially in a textbook, distinguish epithelial cell from muscle cells or neurons, even by their shape, but also huge difference in function. So not only they look different, say epithelial, muscle, and nerve cells, neurons, but they also perform a different function. Not only they express a different kind of genes, different genes are transcribed within their nuclei of epithelial, muscle, and nerve cells. Meaning that there is a very specific pattern. What kind of genes will be transcribed in the epithelial cells? And it's quite different, although somewhat overlapping set of genes will be transcribed in the muscle cell, right? So in other words, this attachment will dictate whether or not a particular type of cell function properly, right? And so what happened due to this mutation in amarin, and if amarin is mutated, there are certain mutations, that the nuclear lamina is not attached properly to the lamin. In some cases, it's just like almost completely separate from the inner membrane of the nucleus. And not only that, it would also affect the ability of nuclear lamina to recognize the correct portion of the DNA. And that this correct portion of DNA will not be transcribed properly. There will be messed up, complete. Certain genes which normally in the nerve cells, say for instance in muscle cells, will not be transcribed, start transcribing, and these cells becoming more like epithelial cells type of, or a certain type of hybrid cell, right? So the function of all of these cells, because we're talking a certain individual carrying mutation in nuclear gene encoding, say for instance, lamin or amyrin, right? Then the function of all of these cells will be affected, and eventually the disorder will develop soon or later, depending on what kind of mutation present in lamin or in amyrin. All right? And the reason behind that is that the nuclear lamina being involved in regulating what kind of genes are transcribed and what kind of genes are silent, not function properly due to mutation in lamin and amyrin, and therefore this transcription goes wrong. Genes which normally must remain silent, they will be transcribed. Genes which normally will be transcribed become silent, and so on. All right? Okay. Any questions, guys? Yeah, this mentioning this free wine, I think it's some inhibitory effect. Or you're just tired, right? The most logistic explanation. If you're tired, just let me know. I'll tell you another joke. Okay. All right. Okay, so now let's talk about some gymnastics. Gymnastics performed by these two membranes surrounding the nucleus by itself. The membrane which forms this nuclear envelope. The outer one exposed to the cytosol, and the inner membrane exposed to the inner space within the nucleus. What is the name of this space within the nucleus? What do you call the space within the nucleus? Sorry? What? Nuclear? No. Matrix? Good. Yeah, it's matrix, absolutely. Don't worry. I realize that most of the people, not only students, they build by this short memory. They have short memory, right? And the advantage of having short memory is the best illustration would be Catch-22. You read? No? Okay. So, the more you know, the more you forget. You know what the continuation? The more you forget, the less you know. The less you know, the less you forget. The less you forget, the more you know, and so on and so on. All right? So that's the advantage of the short memory. Not only that, but not to talk about some emotional things. People would tend to crush out all these unpleasant things and try to be a pleasant thing, but in the night, all these things coming to your brain and blah, blah, blah. But I'm sure that for most of you, it will be not cell biology related. Okay? All right, good. So let's talk about the gymnastics. So the matrix is a space inside the gymnastics is two membranes, right? So first kind of exercises these two membranes do. So far, when we were talking about the nucleus, okay, it's surrounded by two membranes, the outer one and the inner one. All right, so we already know, oh, there are those pores, right? Those pores we call nuclear pore or nuclear pore complex. Now, the important thing to remember is that it's not just a pore and these two free ends of the inner and outer membrane kind of located somewhere in the air. No, they join each other. So it's kind of, they continuous with each other, and they form the structure like that. Within each of these nuclear pore complexes, these two membranes, the inner and the outer membrane, will join each other, right? And then some proteins will be attached to these joint membranes, inner and outer membrane, and they will form these nuclear pore complexes through which all of the communication, import of molecules into the nucleus and export out of the nucleus will take place. So this is the first gymnastics. These two membranes, they actually join each other in a particular space in those nuclear pore complexes. Now the second type of movement is the movement of the gymnastic performed with, by the outer membrane of the nucleus, this membrane, right? The outer membrane of the nucleus. So in certain places, what you will see is that actually this outer membrane from a certain type of invagination into the cytoplasm, right? And this invagination of the outer membrane of the nucleus would create a new organelle with a completely different name, the endoplasmic reticulum. Right? So we are. And so the endoplasmic reticulum is quite different organelle by its function. It performs those functions which are not performed by the nucleus. And also by its protein composition, right? So it will be fair to say that the endoplasmic reticulum by itself is the organelle which not only originates from the nucleus, specifically is formed, the endoplasmic reticulum, by the outer membrane of the nucleus being elongated, folded out of the nuclear space into the cytosol, right? And so therefore, it's a continuation of outer membrane by its kind of very unique membrane for the endoplasmic reticulum. It will contain proteins and lipid molecules in this membrane of the endoplasmic reticulum that are quite different than proteins that can be found in the nuclear membrane. Not only that, we also talk about a different space, right? So this space between the outer membrane and the inner membrane of the nucleus we call what? Perinuclear space, right? Okay, the space within the endoplasmic reticulum, so just what is the difference, right? You just go left and you go right. You are within the same space. But no, this space of the endoplasmic reticulum has a specific name, a lumen of the endoplasmic reticulum. And it has a different name because the chemical composition within this lumen of the endoplasmic reticulum is quite different. And this is ion composition, composition of the proteins, that is the chemical composition of the perinuclear space. So there is a certain type of gauge here, right? Some sort of Maxwell dynamics, which would regulate what kind of molecules moving where. They would just block this Maxwell dynamics, anything which does not belong to the lumen, and therefore need to be restricted from entering the lumen, right? And so, therefore, the chemical composition of this space, the lumen, inside of the endoplasmic reticulum is quite different than the chemical composition of this perinuclear space in the nuclear envelope. All right? So, this seemingly innocent gymnastic of the outer membrane creates a new organelle, the endoplasmic reticulum, essential organelle with quite unique function, different from the function of the nucleus. Okay, now, some other things about the nucleus. So, you already know that, yeah, ribosomes are assembled in the nucleus, and they are assembled not just within the nucleus, but in a specific type of, or in a specific portion of the nucleus. And the name of this portion is the nucleolus, right? So if you look just on electron micrographs of a typical mammalian nucleus within the mammalian cell, what you will see is that, okay, so the two membranes, the outer and the inner, nuclear core complex, all right, we know about that. But most of the space, most of the matrix of the nucleus is filled with this gray substance. And we will talk about what this gray substance is. It's DNA in complex with protein. But what you also will see at least one comparatively large black hole, black in your image of electron microscopical image, right? So this black hole, this large black hole, is the nucleolus, is the place within the nucleus where ribosomal subunits, 40S and 60S, small and large respectively, they are assembled in the nucleolus. Now, very often the nucleolus is called a suborganelle. And it's called for a reason suborganelle. It's not really an organ. Because unlike the nucleolus is not surrounded by any membrane. It's just a complex of many proteins as well as a certain portion, specific genes, a certain portion of DNA encoding a certain component we will talk about. They are located within this black hole. Right? Okay, so the nucleolus, a suborganelle, where ribosomes are assembled. Now, how exactly ribosomes are assembled within the nucleolus? So first of all, remember, all of the ribosomal proteins, all of the proteins, these guys, which eventually will compose the small and the large subunits of the ribosomes, they all are synthesized in the cytosol on those 3 ribosomes. Now,